Okay. I see lots of folks have joined us. So in the interest of time, maybe I'll get started with the housekeeping stuff and I can hand it over to our presenters. So thank you everyone for joining us. Welcome back to the Collaborative Science for Estuaries webinar series. This is Nick Sobral with the Science Collaborative. Today's webinar features a multi-reserve catalyst project in the southeastern United States that compared methods for assessing intertidal oyster reef community structure and ecosystem function. And you'll hear more detail about that project from the two folks who have joined us from that project team in just a moment. But before we get started, I'll give a quick introduction to the reserve system, the Science Collaborative, and this series. So the National Estuarine Research Reserve System is a national network of 30 unique research reserves with more on the way, and they're shown here on this map. This is a NOAA program that works in collaboration with a local place-based partner, either a state agency, university, or nonprofit at each site. Each reserve site includes programs focused on the activities you see on the slide here, which are land stewardship, research, scientific monitoring, training programs for the public and local officials, and education. The Science Collaborative supports science for estuarine and coastal decision makers by coordinating regular funding opportunities and supporting user-driven collaborative research and transfer activities that address critical coastal management needs identified by the reserves. And the Collaborative Science for Estuaries webinar series features project teams supported by the Science Collaborative, program staff, and partners. Speakers share their unique approaches to addressing current coastal and estuarine management issues. And if that sounds interesting to you, feel free to join us for future webinars to learn about new methods to integrate experts and users into the research process and how their research results and products might inform your work. And there are a whole bunch more webinars coming in the next few months, so keep an eye out for those announcements. So before we go any further, just a quick note on informed consent. Webinars are valuable learning opportunities for us as a program. Insights from speakers and participants like you can help us inform how we support others in doing collaborative science. For example, in the development of future workshops, webinars, or resources such as tools, tips, and case studies to add to our guide to collaborative science, which is open access. With that in mind, we just want to inform attendees that insights or feedback you share, for example, in chat or via a webinar survey, may be used for program learning and development. Please note that if we do capture a direct quote from you that we like to use for learning purposes, we will reach out to you and seek permission before we do anything with it and provide an explanation of its use. All such quotes would be used anonymously. We're grateful for the opportunity to learn alongside you. Okay, so quick housekeeping things. All attendees are muted upon entry, as I noted earlier. We'll be handling presentation-related questions via the Q&A feature in your console there. You can enter questions to presenters as they occur to you throughout the webinar, and we'll plan to discuss as many as we can after the presentation period. Chat is visible to all, as I said before as well, so please do use it to talk amongst yourselves, and you can also use it to ask organizers any kind of technical or support questions if you have any. And with that, I will go ahead and introduce our speakers today. So joining us today are Matt Kimball, who is the Assistant Director of the Baruch Marine Field Lab at the University of South Carolina, and Robert Dunn, the Research Coordinator at the North Inlet Winyah Reserve in South Carolina. And I think I'll pass it over to Matt now.
All right, thanks guys. Thank you, Nick. Thanks everybody for coming on. Robert and I are going to share with you the results of our project, but we are only two of a big team that represents 4 reserves and 5 universities, and that we undertook this project from 2023 to 2024. The plan is here, we're going to show you some results, and then at the end, hopefully we get your feedback on what you saw and what you think about the utility of some of these methods. So a bit of background. Oyster reefs are one component of estuaries, which are dynamic, complex habitats with multiple different habitat types, the mosaic or the seascape. Oyster reefs are a prominent component of these, especially in the southeastern US, where many of them are kind of fringing intertidal reefs. You can see from this aerial, you have little intertidal creeks, subtidal creeks, reefs at the mouths, reefs along the creek edge, a dynamic habitat. So reefs come in all shapes and sizes. You have patch reefs and other things, but as I said, we're going to focus on fringing intertidal reefs. And so these are some good pictures of some examples from our research sites. And so these are marsh adjacent fringing intertidal reefs getting right up to the Spartina or right to the edge of the marsh platform. And, they can be vast and expansive, a big, like I said, a big component in southeastern estuaries. We know that in this section of coast, estuaries are influenced by some factors such as tide range and water temperature that can impact the oyster reefs themselves and organismal use of them. So we kind of keep those in mind as we go through this. Here's kind of another example of a fringing intertidal reef with a bit of water behind it. So, a variety of different reef types, but all under the category fringing intertidal reefs. So when you evaluate oyster reefs as habitat, everybody is likely aware of the common methods we do. We use to kind of get at this. Common methods, traditional methods, the oyster biophysical measurements, nectin direct capture. If you've done these before, if you've heard about it, they're pretty time consuming. They can be destructive to the habitat. They can be lethal to the organisms that you're trying to work with, the oysters and the animals that are using them. And, all that kind of comes together to kind of mess up the habitat you're trying to preserve and understand. So people are always looking for ways to get around this, and some developing methods are essentially less invasive or non-invasive and may get at some of these ecological uses of oyster reefs as habitat. They may address or assuage some of the problems with traditional methods as well. We'll talk about that as we keep going. But oftentimes researchers use one type, a traditional method or a developing method, and not necessarily both at the same time. And it's difficult to determine if one is redundant or complementary or better, or just basically see what they are like together. So our main objective was to conduct a side-by-side comparison of these traditional techniques and emerging methods to look at oyster reef communities. And through this process, we wanted to develop a collaborative team in the region that's interested in these topics. And so we used 4 National Estuarine Research Reserve sites spanning North Carolina, South Carolina, Georgia, and Florida. And we used one from each state to try to kind of balance it out. This covers the latitudinal gradient of the southeastern U.S. You can look at these team pictures. We took these each time we sampled. Our sampling took place in October and November of 2023. In October, it was kind of warm, and then it got really cold. And this is one of the kind of important factors of this region. You have this temperature difference, and you have this tide range difference. The surrounding seascapes were similar but differed a little bit. You have some seagrasses up in North Carolina, and you have some mangroves down in GTM, but for the large part, these are Spartina dominated marshes with these fringing intertidal reefs as a dominant component. We focused on 3 reefs in each site, each reserve, and we tried to minimize the variation in these reefs by kind of constraining the characteristics of the reefs we selected, and within the confines of how we could do this. So we have 12 reefs in total, 3 for each reserve. We try to treat them as replicates. It's certainly within the reserve and hopefully among the reserves as well. So for our kind of field sampling campaign at each site, we try to do a 2-day, cram it all in 2 days, and do the non-destructive kind of developing techniques first, and then do the destructive traditional techniques sort of second, so that we wouldn't disturb the habitat while we try to use the developing methods. So for the kind of traditional oyster biophysical measurements, oysters and oyster-associated organisms, these are pictures from the field collections, oyster density, oyster length, cluster height, percent cover, reef height, slope, and rugosity. Then we're looking at, the reef-associated biota like dermo, MSX, mussel densities, boring sponge presence, that type of stuff. So we took these measurements on all 12 reefs. This is a ordination of the reef characteristics, and each of these reserves is a different color. And so there's 12 reefs represented on this. And then we put the kind of important factors, reef characteristics overlaid on this. You can see there's quite a bit of spread on the tidal range and rugosity and reef slope, which are all kind of tied together in density. And that's not surprising. We kind of expected that. A little bit of difference with oyster length and percent cover and oyster cluster height. But for all in all, they're pretty similar. So I think we did a good job with it going forward. And these factors weren't a surprise to us. But the idea is we'll have these kind of multivariate approaches, and we'll start throwing in nectin metrics or organismal metrics or disease metrics into this ordination space to kind of see how that plays out in this, how the assemblages play out, and kind of figure out if these developing methods may be better for one type of reef or one area than another. So that's coming up. For the nectin direct capture, we used traditional methods of fyke nets, fish traps, and crab pots, and we soaked those overnight on all 12 reefs. Followed the same protocol. These are pictures of us deploying the fyke net, retrieving it, crab pots, fish traps, then these kind of common species you might recognize that we collected out in our direct capture. So the assemblage, we collected what we'd expect, the common and key species for these estuarine habitats. About 35 total nectin species and overall. There are some differences between sites, and I think that's largely due to temperatures, water temperatures. When we sampled the southern sites, GTM and Sapelo, the temperatures were in the mid to high 20s. When we sampled North Carolina and North Inlet Winyah Bay, the temperatures were in the teens. So we just happened to be on the cusp of the fall to winter season. But anyways, the usual players. So, then we wanted to get into comparing these traditional methods, these common methods, the oyster measurements and nectin direct capture, to these developing methods. So, like I said before, we set up the order of our field sampling to do these methods essentially, this almost simultaneously with these other methods, but just making sure to do these before we go marching around on the oyster reef or taking oyster samples and whatnot to hopefully have an unbiased sampling with these methods. So for acoustic imaging, we use the adaptive resolution imaging sonar, the NERRS, which is the kind of second generation of the DIDSON. This is a drawing depicting our sampling for that. We would essentially go up to the reef about 5 meters away at high tide when the water's over the reef, put the NERRS in at the surface and kind of view the reef from on top. So the area, the dark gray area, imagine the NERRS is like a flashlight looking down. So we've insonified that whole area. And we would go out there and record for 12 minutes. And with that, we can watch that recording and get a count and a length of all the animals that went through. And so this is the view from a reef, and you can see, I'll orient you to this, with the acoustic imaging recordings, you're looking at it, but you're overhead. It compresses the vertical here. So at the far, around the 4, that's meters, 4 meters away, that's the reef. And then you're seeing fish move through. And on the left, you'll see fish, a school of fish move through, fine. On the right, that's processed. So you'll see red dots and white lines, and the red dots are when we tag that as a target, and the white flashes are lines where we measured their length at their maximal kind of outstretched straight space. So this is an idea of how we process it. We see these fish, we get a count and a length, we record these full 12 minutes. We do it for each minute individually so that we can get some averages. And here's some examples of video on other reefs, but this is detection of larger individuals. And you can see some fish on one side and then a big stingray on the other. And, these fishes, these larger organisms, they likely wouldn't be captured in these direct capture nets or traps. So this is one thing that the NERRS might do better for you. And those are probably red drum on the as the fish. So we can kind of do a little bit of species identification with larger animals based on ecology, but it's difficult to do with everything you see in the recordings. But anyways, this gives you a taste of how we can extract data from these recordings at each reef. And so when we do extract the data from these recordings, it was pretty cool to match up the direct capture counts of total abundance with the acoustic imaging total abundance counts. And so this is just a kind of one-to-one comparison for the 12 reefs. And you can see it's a pretty good similar trend. Nectin counts were similar for both. Their acoustic imaging got about 5 times more animals than the direct capture, but that it followed the same trend as where we got more animals. We saw more detections in our acoustic imaging recordings. So that's pretty good agreement between those two methods. And if you knew what animals were out there, this would be a way for you to sample, perhaps quite a bit more reefs out there. Remember, this is 12 minutes of acoustic imaging. This is an overnight soak of fight nets and traps. So if we look at this is count data for abundance. We look at the length data, you can see the pinkish color is acoustic imaging versus direct capture. The NERRS seems to more effectively sample larger nectin. As you saw from those videos before, we can see those 700 millimeter fish swimming by and record them in our imaging, but they're too fast or too wise to get stuck in those nets. So, we did capture the smaller ones on the acoustic imaging, but also the larger ones. So this is a consideration when using this versus, direct capture. So kind of our second developing method was stable isotope analysis. And we used the samples, the nectin samples from our direct capture collections and investigated food web structure. We used the dried muscle tissue for these, and we focused on a few consumers and a few basal resources. And so for our basal resources, we had 3 Spartina, benthic algae, and particulate organic matter. And we had 5 consumers, but I'm only showing 2 here, blue crabs and oysters. And so with this, these are kind of the isotope values for resources and sites. Carbon shows you the source of primary production. Nitrogen shows you kind of where you are in the food web, and sulfur helps distinguish between these source contributions to consumers. So higher up in these would be consumers, lower down would be producers. We can take these values and then use stable isotope mixing models and kind of look at the primary resources contributing to these oyster reef food webs. And we see that it varies across the South Atlantic Bight or the southeastern US. You have particulate organic matter, the green bars there, more important in North Carolina and Florida. And then you have benthic algae, more important in South Carolina and Georgia. That's the purple. And we think this might be a function of the tide range because it's a bit bigger in the kind of middle of the bight. But we have to do more work on this. But it is interesting. We're seeing differences from these samples and differences between the sites. It'd be interesting to see how that holds up as we look at different types of reefs or different seasons. So we keep that in mind. Another developing method was oyster disease assays. And so we collected 20 oysters from each reef and then did DNA extractions from tissue and then did qPCR to to quantify the presence of dermo and MSX, which are parasites that can have big impacts on mortality of oysters in this area. And there was a high prevalence of both dermo and MSX at all reefs, at all the sites, at all the reserves. This is kind of similar to other studies in the region, so this is not necessarily surprising. If you look at the infection intensity, though, it was low overall, and most reefs were only exposed or lightly infected. And so it seems that these oyster disease may just kind of be kind of an underlying thing, not an important factor. But again, this is just kind of a first swing at this. So for eDNA, we took 2 water samples from over the reef and 2 water samples in the creek channel to serve as like a control called the negative control. And these are 2-liter samples each. We filtered the water immediately or froze the second jar to use for later if we need to. We did this before essentially the same time we sampled with the NERRS before we disturbed with the water just over the oyster reefs, in the thinking that it will have more of a chance of having the organisms that are within the reef, the cryptic organisms, and those also swimming around it. And then we take the creek channel sample to use that as a way to say, like, would we have detected these in the middle without even going near the oyster reef, or maybe perhaps get at the abundance or prevalence of those animals or their DNA in the water. So we were targeting 3 gene regions, COX-1 for metazoans, 12S for fish, and 18S for protists and disease. This is kind of a way to investigate the utility of all these 3 different types of gene regions for detecting species. We're amplifying each gene region 4 times for each sample. There's a ton of DNA sequences per sample per gene, more so than we expected. So it's taking a bit longer, and we've had to add computing power to get through this. But we've got through some of it. Like I said, it's well above normal. We couldn't just use standard bioinformatics because the data sets were so large. We couldn't just send them, blast them to GenBank like normal. But the preliminary analyses for COX-1, we did identify common species such as white shrimp, blue crabs, blenny, goby, and mullet, which are kind of a mix of species that would be around and within the oyster reef. So that's good. And we're continuing to work on the bioinformatics analyses for 12S for fish and 18S for the protist and oyster disease. But we have this idea for analyses, and I'll step you through this. But this is kind of a way to look at eDNA versus the direct capture nectin. And so on the left is a list of all this species, all 35 species of nectin that we collected at all the reefs combined. And then each column represents a reef at a reserve. There's only 2 columns for Sapelo Island because the samples were lost or were damaged before we could deal with them for that one. So there's only 2 reefs at Sapelo Island. So the categories, I'll work from backwards. The red is not caught or detected, and that's the largest category here because not all species were collected at each reef. So that's to be expected. The yellow is only detection in the eDNA. The blue is only detected with direct capture traps, and the green is detected with direct capture traps and eDNA. And so you can kind of see that we did get some, there's some detection with both. There's some detection only with eDNA. For instance, eDNA got the cryptic fish like the goby and blenny that would be within the oyster reef that we just really wouldn't be that good at catching with the nets. They detected blue crab and white shrimp, but not very much. And that's something we're curious about. Crustaceans have hard exoskeletons. They're not sloughing off a lot of slime or DNA, so maybe it's harder for this gene region or other gene regions to detect them. We did get some normal species or common species like mullet and other things like that on here. So this is promising. We did not know or expect anything with this. Like I said, we're trying this for the first time. And so it'll be interesting to make similar kind of heat maps and diagrams with the next two gene regions, particularly 12S, which we've tried and had some trouble with, but we're hopeful that one will give kind of more detection of some of these finfish. And it seems like, at least from this, that eDNA is not a whole substitute for direct capture. It may be a complement, but we'll have to wait and see about these other gene regions. But it does have some positives, like I said, potentially able to collect some of these more cryptic species and show them, to be there if we collected them or not. This was a collaborative catalyst, so a large part of it is our kind of collaboration and outreach and working with end users and addressing current management needs. So, our team got together and started working in 2022 because we all kind of knew each other or heard of each other, and we had collective shared interest about oyster reefs and nekton use in the Southeast. And so we met together before to kind of write the proposal and decide on our ideas. And then as part of our work, we did 2 in-person team workshops, one at University of South Carolina, Beaufort Reef, or the BMFL, and then one at USC Beaufort. And then towards the end, we wanted to get feedback from kind of agency folks and end users and land managers, natural resource managers. So we had a virtual webinar in the last weeks of our project, where we had representatives from 17 organizations, where we shared more preliminary results than what I'm showing you today. But enough for them to get an idea and see what they might do and tell us what their respective institutions or agencies might do with this. And the kind of distilled down output from that meeting was that there was interest in examining more reef types, so not just fringing intertidal reefs, not just natural reefs, perhaps restored reefs, reefs of different ages, harvested reefs. The reefs we were on were largely unharvested. Sort of different settings that reefs were in and stuff like that. Also, we only looked at one season, kind of the end of the fall, start of winter, and so there was some interest in looking across all seasons and seeing if things patterns and other things change as we get to different seasons. Does that, for instance, direct capture count and NERRS count relationship still stay pretty solid, or does that change? Also, from a management perspective, there was interest in us targeting key nekton species, sport fishes like redfish or spotted sea trout or things like that people associate with reef habitats. And that might be something that's possible to do with the eDNA or the NERRS. Just thinking of the preliminary results, we can see some of those larger fish and go from their body shape and behavior and determine what they might be. So very promising, and a real advantage of our collaborative team and our effort to get the word out. And it was good to get that feedback, and we're going to use that to inform future proposals from our group. And so this presentation today is kind of a next step for our project team as well, because we wanted to share these results with the Science Collaborative and Reserve System and affiliates, and kind of continue the discussion about the pros and cons of using these methods. And then, we're always or currently working on writing papers and reporting results. And like I just mentioned, we're going to develop a science collaborative full proposal that's in the works for this next RFP cycle, where we hope to expand into those different reef types. We hope to expand into all seasons, and we're going to include all the reserves in the southeastern US, so up to 5 reserves. So it'll be a pretty comprehensive thing over multiple years, but we feel pretty confident that our kind of preliminary pilot results from this catalyst show some promise, and we're all a bit eager to try them out in more environments. So that was a quick, I'll say, 30-minute run through some preliminary results. Now, I wanted to stop and see if any of you all had any questions on what we just showed you. And we can go back and go through the slides and whatnot and get to specific things, but we have time, and this is the input that we're most interested in. So if anybody has any questions, you can unmute and talk, or you can go into the chat, or whatever works best for you. And Robert and I are both going to kind of field these as we can.
So Matt, we've already got some good questions in the Q&A box, mainly about NERRS and eDNA. I'll encourage the other members of the team that are on the call to jump in as well if there's a particular question that you feel like addressing. Why don't we just start with the first one, which was from David. Did you use NERRS to image the shore or the bare shore without oysters? Some sort of control comparison for the acoustic imaging study.
We did not for this one, but I have done that in previous studies for comparison. We just did not. We were trying to just go through very efficiently and just focus on oyster reefs. But it's always good to have a control like that. And we are definitely going to plan for that for the next. One problem we have is sometimes these fringing reefs are huge and line the creeks forever. So finding a bare spot so far away may not be, applicable to what you're looking at with the reefs.
Another little aside to that is thinking about the processing cost and the processing time associated with these imaging files. There's a bit of a trade-off between, collecting your data in situ using nets and traps, where you're out there all day. The NERRS is easy. We can sample multiple reefs in a 2-hour high tide window, but we then spend weeks in the lab running through those imaging files over and over again.
Yeah, and I'll also say there's ways for us to do that quicker by subsampling. We looked at our whole 12-minute blocks, but we've done this before where we subsample, 2 minutes or 3 minutes out of a 5-minute block, or you can even go frame by frame, just depending on how much you want to get out. And those patterns, there's some papers that say that those patterns you see in subsampling are essentially the same patterns you see if you do the whole block. So there's ways to work around that, but it's a good point.
Really quickly, get a couple more NERRS questions, and then we'll jump to some eDNA questions. So John asks about the cost of the NERRS hardware and software, and then another question was about the impact of turbidity on using acoustic imaging.
So the cost is real. That the setup we have costs about $120,000 10 years ago. I don't know what it costs now, but you get the software for free in life. They just keep giving you updates. It's very easy to use. So once you have the initial input to buy the thing, you should be pretty good. Ours is, we found it's very robust. We treat it nicely, but we still take it in the field, and we haven't had any troubles other than ways I've broken it. You have to fabricate some mounts. The drawing you saw, we used a boat mount on the gunnel. We also have a stationary mount that's a tripod. These are pretty standard. I've fabricated multiple of these over the years, not more than several $1,000s to get both those set up. It's pretty straightforward. You have a laptop, and then you can roll with it. And the learning curve on the software is very very quick. We found we've had folks that have never used this before really get into it in a couple days, and so it works quite well. What was the second question, Robert?
How acoustic imaging is useful in a turbid environment.
Oh, yeah. Turbidity, like sedimentation, doesn't really mess with it so much, but air bubbles and stuff like that really get to you, and you can't. You'll just see kind of clouds. But we would get up on site. I didn't mention this part. We would get parallel 5 meters away from our reefs, put 2 anchors in on each side, caging anchors, so it wouldn't disturb as much, and turn the engine off. And so we were not disturbing the surface, not making as much noise as possible. And then we just kind of sit there in silence while the thing runs. And it runs by a generator that's on deck, which you can pad to make it not so bad. So in these types of settings, I think turbidity is negated. And you really wouldn't be out there on a day where it's really churning and chopping. So it seems to be okay. But in more anthropogenic settings where you're looking at water control structures or like revetments or something, that may be something you have to consider.
Another question we often get is about the resolution of the acoustic imaging videos. There's some settings on the NERRS system you can tinker with to change the resolution of your video, but we were hoping or considering potentially using the acoustic imaging to actually get an estimate of the reef complexity. Maybe we could trace the outline of the clumps on a reef, for example. With our setup where we're anchoring the boat, and so we're still rocking just slightly, we're really not able to get high enough resolution for that. Before, mobile nekton, so fishes swimming around, we're pretty confident that we're able to see things as small as a couple centimeters. And so that's really why we're able to get such high numbers from the NERRS counts, because we're in a 12-minute recording, we would regularly be enumerating over 1,000 animals.
Yeah, and that's the advantage of the NERRS over the Didson. The resolution is twice as high, but it's 3 megahertz versus the old one, which was, I think, 1.8 or 1.2.
So let's jump to maybe some more info on the eDNA bit. Questions about, is eDNA still complementary to traditional capture? Why it was sampled on the flood tide instead of the ebb tide? Sampling eDNA at night. Mercer, if you're interested and want to get on video, please join us. I see you're answering in the Q&A box. That's helpful too. I'll just briefly say that we have looked at the negative control samples. Someone asked about the sort of creek channel versus on reef samples. We designed the study to be able to use a negative control in the eDNA on that eDNA question. We did see some differences in terms of the species that were detected on reef versus in the channel, which we expected. We haven't been able to really dive into those data just yet, so that's still more to come on that.
Is Mercer on?
He's answering in the chat. If anyone wants to come off of mute and just ask a question directly, that is fine as well. Some useful comments here as well from Jeff and others about the various gene regions.
Hey, Robert, this is Mercer. Can you hear me?
Hey, Mercer,
Yes.
go ahead.
Excellent. Sorry, we didn't have the ability to access audio or video, but the ability for audio just popped up. So I want to say thank you for doing that. So yeah, to answer some of these eDNA questions, for why flood versus ebb, the thinking is the tide comes in and starts flooding the reef, things are going to be opening up, they're going to be unburrowing, unhiding, and they're going to start the feeding process, releasing mucus and skin cells and all sorts of goodies that we can pick up in the water sample. So that's the thinking there. But, the more I think about it, assuming money wasn't a limitation, looking at the ebb tide would also be interesting to see if we get a different suite of organisms as things are closing up, as the water is leaving or burrowing or what have you. But we chose the flood just because things are coming out to feed, and they're being messy doing that. In terms of reef versus middle of channel, we were looking for presence absence. But if something was found on both the reef and in the middle of the channel, the latter being our negative control, we are just doing a read count. So let's say we got 100 DNA of our 1.5 million DNA sequences that we got from that sample. If 100 were blue crab and those were on the reef, and we only got 5 blue crab sequences off the reef, we have some level of confidence that that blue crab was associating with the reef. Yes, also in the system, but probably associating with the reef. So kind of doing a comparison of reads or DNA sequences. So the heat map that you saw was for cytochrome oxidase 1, the COX-1 barcode of life, which picks up all multicellular animals, and so that's the gene region for which we have the best data right now. 12S that we're targeting for the fish, the fin fish, the sport fish. We are just getting that data, so we don't have anything to show you just yet. And in 18S, which is not a mitochondrial gene, nuclear gene, we're going after the protist. So hopefully we'll know more about the eDNA very soon. Anything else I needed to answer, Robert or Matt?
No, I think that generally covers most of them. Thanks, Mercer. Appreciate it.
Thanks, Mercer.
Let's see. There's some other interesting ones in the chat. Feel free for others to come off mute if you'd like. There's some questions about sort of management and which of these techniques might be particularly useful in various management contexts, or what were the management questions that we were really addressing with this project. That's a really good point, and that's actually the goal of our next larger 3-year proposal, is to address more targeted management questions using some of the techniques that we tested here. So we heard from state natural resource management agencies and NGOs doing restoration of oysters that they wanted to see some of these techniques deployed at different seasons for multiple years and across different reef types and under different environmental conditions. And so deploying a given method for sampling is really going to be dependent on the local management need in the place where you're doing the work. So I wouldn't do anyone any favors to sort of make a broad brush statement about what we are aiming for here, but we think that some of these are definitely going to be useful under different contexts. Zach asked about which techniques are particularly useful when, and that's what we're actually hoping for the ordination analysis to tell us some more about. We can characterize the biophysical structure of the reefs across 7, 8, 10 different parameters, and then look at how some of these nectin response variables overlay on that ordination space. And so we're hoping to be able to make some recommendations using that analysis. Let's see. I see a question about able to assess spat recruitment with NERRS. No, the resolution's really just not that high. If you're looking for non-invasive recruitment estimates, you really just need to put out settlement plates or shell string collectors for spat.
Yeah, but we may be able to get at some of that slope and rugosity and stuff like that if we were able to position the NERRS in a certain way. What we showed you was the horizontal. If you put it in the vertical, you might get that kind of mountain range looking outline that we can compare. But that'd be a whole nother test to see how it compares against those traditional measurements.
So I see one more interesting question here from Dorothy that I'd like to address, and then we're going to pose a few questions to y'all. So Dorothy teaches natural resources to high school students, and she asks which of these methods would we suggest that she helps her students learn about or learn how to use for folks interested in careers in this field. Matt, do you have an opinion on that?
Well, I think it's always fun to kind of delve into these emerging methods and look at those and provide your fresh perspective to it. I've had an interest in applying the acoustic imaging for a while now, and it's been fun to put them kind of put it to the test in different habitats in different places and see what you learn and develop the technique even further. You're essentially testing it for the manufacturers. And that's kind of what our whole project is doing with this. So if you're curious in this arena, I would kind of understand the basic ecology of these systems and then say, like, how can we get a little bit more of a detailed answer out of what we've been doing? And that's where these kind of emerging methods become of interest to me. And we're applying. I'm most interested in acoustic imaging. We're applying this in estuaries. There's not a lot of activity in that realm, but they've used it all over the world, especially in deeper water and other places to do a lot of cool stuff. So it might get students interested in exploring that elsewhere. Also, from an education standpoint, it's interesting to have things where you can watch digital recordings of it and analyze it from afar. So maybe you can expose more people to that by doing exercises in the classroom that eventually may lead to field stuff. So that's one thought. Robert?
Yeah, I'll provide just a contrasting opinion, which is that I think probably students need to get out in the field and handle some fish and some crabs. These emerging techniques are terrific and provide some amazing information, but you really need to have the basic understanding to be able to interpret what they're telling you.
Yes, no substitute for that. I agree.
Thanks a lot for all of these questions. That's a really useful set of discussion. Nick, could you push the poll question to everyone? So we're gonna just have a brief interactive session here for the last 5 or 10 minutes. You should have just received a poll question asking what are the top management needs related to oyster reefs in your favorite estuary or wherever you work. We should be able to see the results here if everyone would give us a vote here over the next 30 seconds. Nick, I can't see anything about the results, so
Yeah, I see it. Keep an eye on it.
you'll have to let us know when we're ready to.
We're at about 75%, so most people have taken it.
Great. All right, we can cut it off.
Sure.
All right. Yep.
All
Great. Thank you. Interesting. So a lot of restoration, water quality and habitat provisioning coming in second. Meredith says shoreline protection. And we've deliberately put water quality in sort of vague terms here. I mean, that could be water quality is problematic, and so oysters can't be harvested from a given area, or that might be, an interest in increasing oyster abundance to remedy some water quality problems. All right, thanks. So with that in mind, we're interested in hearing about from you all if any of these techniques specifically or the ecological information that they provide are particularly applicable to your work in, say, other habitats beyond oyster reefs, or just would be of particular interest in your estuary. And feel free to use the chat or jump in directly if you have a comment. Jay mentioned that ecosystem service evaluation would be another useful tool in the toolbox to understand the return on investment for restoring reefs.
Yes, that's for sure one thing we've been thinking about, like, when do restored habitats and was restored reefs reach equivalence to natural habitats? Is there the initial bump in production, or is there, how long does that take? Or also the different types of reef restoration, because they do different types more so in some states versus others. And so we have kind of a natural gradient to examine that as well.
Yeah, I'm seeing lots of comments about habitat restoration assessment and effectiveness. So, non-destructively sampling a restored reef is certainly a nice use of these non-invasive techniques.
Yeah, and it certainly seems that you could use these non-invasive techniques to sample many more reefs of all these types in the same time period that you could sample a few reefs with the traditional methods.
How about this next question here for y'all? Are the reserves in your area or the natural resource managers in your state interested in understanding food web structure and the use of oyster reefs as habitat, or are they typically thinking more about oysters as a fishery resource? Some resounding yeses. Well, that's great. But more so as a fishery resource. Yeah, I mean, I think that's probably common. Yeah, we actually had the Billion Oyster Project on our fall virtual webinar, so they are familiar with our work here. Last question is kind of general. Are there research questions that are applicable to management in your estuary, mainly related to oysters that we haven't discussed today, but might merit some research in your eyes? Brendan mentioned the use of eDNA for invasive species. I think that's also sort of relevant to this question. Subtidal reefs, yep. There's definitely been some work on subtidal reefs using NERRS in North Carolina, and so this technique is definitely spreading.
Yeah, and on artificial reefs as well.
Mm-hmm.
And it's been pretty promising.
All right. Well, thanks again, everyone, for joining today. We're just about out of time. Happy to stay on and answer any other questions that folks have. Matt, do you want to go to the next slide and just, yeah, so here's Matt's contact information. Feel free to reach out to he or I if you'd like more information about, or any of the other team members if you'd like more information about our project. And keep an eye out for the papers that we plan to put out here in the next year.
Yeah, and the National or the Science Collaborative maintains web pages for these projects. And if you just look it up, this web page will have links to products that we already have completed and future products, so papers, past like handouts and posters and anything that's related to it. So, and I believe they maintain these for quite some time. Also, the data will be available after an embargo period, so that will be downloadable for people to explore. And that includes the NERRS videos and the software and, everything we just showed you.
Thanks, folks. As we're wrapping up here, I'll just make a note that you can also provide any questions that didn't get answered that you still are, or that occur to you after the fact via the exit survey. So the exit survey will launch automatically as soon as you exit the webinar session, and it'll also be sent tomorrow at the same time as the webinar in a follow-up email. And that follow-up email will have the link to the summary resource page that'll have at least a placeholder link for the YouTube video while we process it. Usually we have it ready within a few days of the presentation, but sometimes it takes a couple days. So just keep an eye on that. And any other similar resource materials will be there as well. So thank you to everyone for joining, and thanks for such an active and engaged Q&A session. That's always good to see. Lots of questions flying in. We'd love to see that. And keep an eye out for future webinar announcements. So with that, everyone enjoy the rest of your day.